Hey, I'm Dr. Tisha Vyas. I'm Assistant Professor of Biotechnology, Siusha Institute of Science located in Ahmedabad, Gujarat. I'm Doctorate in Biotechnology. Today, I'm going to talk about the structure of Eukaryotic Flagella and Cilia. The content of this video is Introduction, Definition, Types, Structure, Importance, and Functions of cilia. Cilia are fine hair-like movable protoplasmic processes of the cells, which are capable of producing a current in the fluid medium, helping in locomotion as well as passage of substances. As we can see, flagella are longer but are fewer. Only 1 to 4 flagella occurs per cell. Example, many protists, motile algae, spermatozoa of animals, bryophytes, and pteridophytes, choanocytes of sponges, gastrodermal cells of coelenterates, zoospores, and gametes of thallophytes. Compared to flagella, cilia are smaller but are numerous. They occur in group ciliata of protista, flame cells of worms, and larval bodies of many invertebrates. The cilium came from the Latin word meaning eyelash. The plural is cilia. It is an organelle found on eukaryotic cells in the shape of a slender protuberance that projects from the much larger cell body. In eukaryotes, motile cilia and flagella together make up a group of organelles known as untulipodia. Eukaryotic cilia are structurally identical to eukaryotic flagella, although distinctions are sometimes made according to function or length. They occur in a group ciliata of protista, flame cells of worms, larval bodies of many invertebrates, epithelium of respiratory tract, renal tubules, oviductal funnels, etc. Cilia are also present on tracheal and bronchial epithelial cells, specialized to send back the dust particles into the pharynx so that the lungs remain unharmed. However, cigarette smoking reduces as well as it stops ciliary activities so that airborne dust particles passes into the lungs of smokers, causing irreparable harm. Both cilia and flagella are structurally similar and they possess similar parts, that is, the basal body, rootlets, basal plate, and the shaft. Cilia can be divided into 3 types: primary cilia, also known as the non-motile cilia, the motile cilia, and the nodal cilia. Primary cilia. In animals, non-motile primary cilia are found on nearly every cell. In comparison to motile cilia, non-motile cilia usually occurs 1 per cell. Nearly all mammalian cells have a single non-motile primary cilium. In addition, examples of specialized primary cilia can be found in human sensory organs such as the eyes and the nose. The outer segment of the rod photoreceptor cell in the human eye is connected to its cell body with a specialized non-motile cilium. The dendritic knob of the olfactory sensory neurons, where the odorant receptors are located, also contain the non-motile cilia. Motile cilia. Larger eukaryotes such as mammals have motile cilia as well. Motile cilia are usually present on the cell's surface in large numbers and beat in coordinated waves. In humans, for example, motile cilia are found on the respiratory epithelium lining the respiratory tract, where they function in the mucociliary clearance of sweeping the mucus and the dirt out of the lungs. In female mammals, the beating of cilia in the fallopian tubes moves the ovum from the ovary to the uterus. Nodal cilia. Nodal cilia are only present in the early developing embryo. The nodal cilium is of similar structure of the primitive cilium in having no central apparatus, but it does possess the dyname arms that enable it to move or spin in a circular direction. The spin is clockwise, and this causes a flow of extra-embryonic fluid to move across the nodal surface directed to the left. Primary cilia around the nodal cilia senses the directional flow which activates the nodal signal, establishing left to right sideness. Now, let us have a look at the structure of eukaryotic cilia. In cilium, the sheath contains a number of thick hairy outgrowths called flimers. Internally, it contains a semifluid matrix having an exoneme of 9 peripheral doublet fibrils and 2 central singlet fibers. This type of arrangement is called 9 plus 2 or 11-stranded. However, 9 plus 1 and 9 plus 0 arrangements have also been observed. The 2 central singlet fibers are covered by a proteinaceous central sheath. They are thereby connected by a double bridge. Each peripheral fibril consists of 2 microtubules, or also called the subfibers B and A. The subfiber A is slightly narrower. It bears 2 bent arms, the outer one having a hook. They are about 15 nanometer long and made up of protein dyname with ATPase activity. Such activity is also present in central fibrils. The movement of flagella or cilia occurs due to sliding motion in which dyname arm establishes temporary connection with subtubule B of adjacent doublet fiber. The peripheral doublet fibrils as well as central singlet fibrils are made up of tubulin. Each subfiber or central singlet fibril contains 13 protofilaments. The peripheral doublet fibrils are interconnected by A and B linkers of protein nexin between B subfiber of one and inner side arm of A subfiber of adjacent fibril. Each of their A subfibrils sends a radial proteinaceous column to the center. It is called a spoke. The spokes are broader internally to form head or knobs. Head is connected to central proteinaceous sheath through transition junction. The cilia and flagella move by sliding of the doublet fibrils against one another. They get the energy from ATP. Now, let us see that how the cells as well as the bacteria, they can move with the help of cilium. In cilium, they are having a number of flimers, the undulatory wave moving down from the base to the tip it also pulls the cell along instead of pushing it. There is always a power stroke and a recovery or return stroke. The power stroke is able to move the fluid with a jerk in the direction of the stroke. The cell moves in the opposite direction if it is immotile. The recovery or return stroke is slow and without much force. Therefore, it does not cause much disturbance in the fluid medium. The rate of ciliary movements is 10 to 40 strokes per second. Now, let us have a look at the functions of eukaryotic cilia. The dynein is the exonym formed bridges between the neighboring microtubule doublets. When ATP activates the motor domain of dynein, it attempts to walk along the adjoining microtubule doublet. This would force the adjacent doublets to slide over one another if not for the presence of nexin between the microtubule doublets, and thus the force generated by dynein is instead converted into a bending motion. It also has another functions like sensing the extracellular environment. Some primary cilia on epithelial cells in eukaryotes act as cellular antennae, providing chemosensation, thermosensation, and mechanosensation of the extracellular environment. This cilia then plays a role in mediating specific signaling clues, including soluble factors in the external cell environment, a secretory role in which a soluble protein is released to have an effect downstream of the fluid flow, and mediation of fluid flow in the cilia are motile. Some epithelial cells are ciliated and they commonly exist as a sheet of polarized cells forming a tube or tubule with cilia projecting into the lumen. This sensory and signaling role puts cilia in a central role for maintaining the local cellular environment, and maybe why cellular defects cause such a wide range of human diseases. I hope you got enough information on eukaryotic flagella and cilia. Thank you.